that's not working. So the cell body, it's labeled right here in the picture. So that's that part right there. That's the site of the nucleus and all the different organelles in the nerve cell. It has a well-developed rough endoplasmic reticulum and Golgi bodies because the nerve cell, it makes a lot of proteins. Because the main function of the nerve cell is to be sending signals, these proteins are the ones that are actually sending the signals. And specifically, those proteins are called neurotransmitters, but we're going to talk about that in a second. The next part is the axon. The axon is pretty much this long part of the cell right here. So what the axon does, it actually conducts the signal. In fact, the signals actually originated at the axon, and eventually that signal is sent to the axon terminals, and neurotransmitters are released there, and that's how the signal spreads throughout your nervous system. So the axon terminals, that's specifically the secretory region of the nerves. So that's where there's a exocytosis, and it actually releases the neurotransmitters there. Other names for the axon terminal are the synaptic knob or the bouton. Now the dendrites, those are back right by the cell body in that picture. So this is the region where you are actually receiving the input. So this is where the neurotransmitters that are sent from the axon terminal, those are received at the dendrites and processed and sent to the cell body, and then through the axon, and then it repeats itself all over again. And the dendrites, it's important that they're branched because that actually increases the surface area for the reception. Now, you also have myelin sheaths. Those are pretty much covering the axon intermittently, and the spaces between each of the sheaths, that's called the nodes of Ranvier. And the purpose of, so right here in this blank, the gaps are called nodes of Ranvier. That's Ranvier. Type it up. Okay, so the purpose of the myelin sheath, it's to speed up conduction by insulating the nerves in intervals. So the intermittent insulation, it causes the action potential to be able to jump from one node of Ranvier to the next, and essentially it speeds it up. And right by the nerve cells also, there's also such things called Schwann cells, and Schwann cells that actually makes the myelin sheath in the peripheral nervous system by wrapping around the axon. Another thing, oligodendrocytes, that's pretty much the central nervous system analog of Schwann cells, and it makes the myelin sheath for the central nervous system axons. So in terms of the nervous system, there's 3 different types of neurons. I don't know if I have it later in these notes. Let's see. Doesn't seem like it. So write this down on the side, I guess. So 3 different types of neurons that you have. You have sensory neurons, you have motor neurons, and you have interneurons. So the sensory neurons, those are the ones that are sending signals to the central nervous system. The motor neurons, those are sending signals from the nervous system, from the central nervous system to the rest of the body. And the interneurons are the specific neurons that are actually contained within the central nervous system. So, like I don't think I said earlier, but the central nervous system, that is your brain and your spinal cord. Okay, so the next part is the synapse. So the synapse is actually the space in between the two different neurons, and that's where the conduction actually happens from one cell to another. So the axodendritic synapse, that's the axon terminal of one neuron, which is the presynaptic neuron, and it's between the presynaptic neuron and the postsynaptic neuron. Whereas the axosomatic synapse, it's between the presynaptic neuron and the postsynaptic neuron. Hold on. Okay, so the first one, it's between the axon terminal of one and the dendrite of the other, whereas the second one, it's in between the axon terminal and the cell body of another. That's how they're different. And then you also have axonic synapses. Those are a little bit rare, and they're going to be between the axon terminal of a presynaptic neuron and the axon hillock of a postsynaptic. Okay, so this picture right here, it illustrates how synaptic activity actually works. So what happens is you have an influx of calcium into the neuron, and that causes the action potential to be spread through the neuron. And the calcium, it causes the neurotransmitters actually to be released into the synapse, and then it'll be taken up by the next neuron. So the dendrites of the next neuron will pick up those neurotransmitters, those signals, and then pass those along. That signal will get, the action potential will continue to get passed along. So the transmitter molecules does, I mentioned a few times. Does anyone remember what the name of those are called? The actual proteins that are sending the signal. Go ahead and type it in the chat box. So here's a hint. The actual name of the transmitter molecules is in the next line when you're describing the action, where the action potential is being described. Yeah, that's right. So the transmitter molecules, they're called neurotransmitters. So that's pretty intuitive, I guess. So the action potential, what actually happens in the action potential, or I guess the definition of action potential is when there's a release of neurotransmitters by a presynaptic axon terminal, and then they're picked up by the receptor of a postsynaptic neuron. And the action potential, obviously, that's the signal being sent out. In the way that the release of the neurotransmitters work, what happens is there's an exocytosis of vesicles that are containing the neurotransmitters, and it's triggered by a calcium influx, as you can see in this picture right here. So when the calcium is taken into the cell, that's when the different vesicles in there with the neurotransmitters, those are exocytosed. Okay, so neurotransmitter reception. So the way that works is the neurotransmitters, they're actually diffused across the synaptic cleft. They bind to receptors on the dendrites, and that opens up ion channels that causes a change in membrane potential of the postsynaptic neuron. And if this potential is great enough, then it'll trigger a full-fledged all-or-nothing action potential in the postsynaptic neuron. So I know in that little description right there, it sounds a little bit confusing, but we're gonna go over action potentials in more detail in just a second, and I think it'll be a lot easier to understand. Also, neurotransmitters are they're quickly eliminated from the cell and from the synapse as well. So they're taken up by the presynaptic terminal, or they diffuse away and they're destroyed by enzymes as well, so that they're not persistently stimulating the postsynaptic neuron. So like one example is so with cocaine, what cocaine does is it actually blocks these reuptake inhibitors, and because of that, your neurotransmitters are stuck in the synapse, and they keep getting received by the dendrites, and they're not actually reuptaken, but rather the signals just keep on going on, and that's why it's pretty much like a super stimulant. Okay, so some examples of neurotransmitter molecules I have listed right there. Acetylcholine is one of them, norepinephrine, dopamine, serotonin, histamine, and ATP. Those are all different types of neurotransmitters in your nervous system. Okay, so the synaptic knob, we talked about it a second ago. That's just another name for the axon terminal. And the synaptic knob, it contains vesicles of all the neurotransmitters that are waiting to be exocytosed or sent out of the cell. Whenever the action knob, it causes an influx of calcium, which we already talked about, and that signals the vesicles to fuse with the cell membrane and be exocytosed, and therefore releasing them. So fatigue is what happens when you have continuous synaptic activity, and because of the continuous synaptic activity, you end up having depletion of the neurotransmitters, and then you end up having fatigue. All right. So in action potentials, they're all or nothing. So that means if the membrane potential reaches the right amount, then the full action potential will be sent. If not, it's not going to be transmitted. So that's why it's called all or nothing. So that blank right there, it's all or nothing. And the blank right before that, I don't know if I made it clear, but that's calcium, an influx of calcium. Okay, so now we'll talk about the resting potential. We'll look at this action potential graph. So the resting potential, it's heavily influenced by the NAK plus pump, which is also called the NAK plus ATPase. We talked about that, I forget, a couple days ago, one of our other classes. So the way that works, it sends 3 sodium molecules out and 2 potassium molecules in. So because you're sending out more positive molecules than taking in, the net membrane potential inside the cell, it's going to be negative, and it's going to be net positive on the outside. And there's also, with the NAK plus pump, there's a little bit of K plus leakage, so that makes it even more negative because, first of all, you're only taking 2 K pluses in, and some of them are leaking out, so the positive charges are leaking out a little bit, so it makes it even more negative on the inside and more positive on the outside. The resting potential for our neurons, it's negative 70. So negative 70 MV, because the cell, like I said, is going to be more negative on the inside. Now, the gradient that's, whoa, I'm not able to scroll through this document properly. Just see this. There we go. So the gradient that's created by these charges, it's called the electrochemical gradient, and that's the combination of electrical and chemical gradients. Because you have a chemical gradient because of the sodium and the potassium molecules that you have there, and you also have an electrical gradient because of the charges. Any questions so far? Just type it in the chat box. So now we'll look at this actual graph of an action potential and just really discuss that in a little bit more detail. So we have a few different stages of an action potential. Let's see if I can get all of this on the screen. Okay. So the first stage of an action potential, it's called the resting stage. So this is where the membrane potential is going to stay at negative 70, and the pump, it pretty much maintains that, and the sodium on the outside and the potassium on the inside. The ion channels are going to stay closed here, so that the established ion gradient, it doesn't leak as much. I mean, you still have the potassium leakage, but it tries to minimize that during the resting stage. The second stage, it's called oh wait, I just saw in the chat box you said not really and no. What are you guys referring to? Oh, okay, got it. Yeah, so the second stage, it's called depolarization. And in depolarization, what happens is, as you can see in the picture, the sodium channels open. So sodium is going to rush into the cell. And because the sodium is positively charged, the membrane potential is going to shoot up. And you can see that it eventually reaches plus 30. So it goes from negative 70 to plus 30. And at this point, there's a lot of sodium on the inside, and there's still a lot of sodium on the outside because the other part of the pump that's pushing the potassium out, it's still closed. And then the third stage, it's called repolarization. In repolarization, what happens is the potassium channels open up, and then the sodium channels close. So what happens is now that the sodium is still stuck inside, but since the potassium is open now, it's going to rush to the outside, and the membrane potential, it's going to drop back down. Now the fourth stage, what happens is because the potassium channels, they don't close fast enough. There's going to be an overshoot of the potassium that's going out. So it actually, your membrane potential, it goes past the resting potential, the resting membrane potential. So it goes even more negative than negative 70. And this stage is called hyperpolarization. And finally, the pump kind of fixes itself, and it sends sodium out and keeps the potassium in there, or whatever potassium is now left in the cell. It's going to keep it in there, and it's going to pretty much bring the resting potential back to negative 70 and back to that resting state. And if you look at the top of that diagram, it says absolute refractory period and relative refractory period. So what that's referring to is, let's see, this fifth blank over here is going to be refractory period. And what that's referring to is the fact that since the sodium-potassium pump is working to reestablish that original resting state, the cell's not going to be able to process another action potential. So during the absolute refractory period, it definitely can't do it. In the relative refractory period, depending on how strong the action potential is, it might be able to do it. So during hyperpolarization, a neuron can still transmit an action potential, but it would have to be a really strong action potential. It would have to be a really strong signal. So a lot of neurotransmitters would have to be involved there. Whereas in absolute refractory period, no matter how many neurotransmitters are being transmitted, there's going to be no action potential being made. Okay, so that is the action potential. Scroll down a little bit. This mouse is really shitty. Okay, so I already mentioned the whole all or none nature of the action potential, but I'll talk about it a little bit more. So it really depends on a threshold. So in this picture right here, you can see in that graph, there's a little dotted line that says the threshold. So unless the stimulus or the action potential, it depolarizes above this threshold. So during this depolarization stage, unless it goes above the threshold, then an action potential will not occur. And if it does, then it will occur. So that's why it's all or none. So pretty much all of the action potentials will have the same magnitude. So I know earlier I said in the relative refractory period you need a stronger action potential, but I'm really referring to the concentration of the neurotransmitters that are being transmitted from one neuron to the other. Okay, so I think this is a little bit redundant, talking a little bit more about the sodium-potassium pump, but we already went over that. 3 sodium out, 2 potassium in, net positive out, so inside the cell negative, outside the cell positive. And another thing that's not written there, it uses energy, so it's called an ATPase. Now, within your nervous system, you have both excitatory and inhibitory nerve fibers. So an excitatory nerve fiber, it stimulates the action potential to occur. And within those, or between those excitatory nerve fibers, you have excitatory synapses. And over there, you have receptor binding that cause the postsynaptic potential to be more positive and cause depolarization. And if it gets above that threshold, the action potential results. Whereas the inhibitory, the way that works is it inhibits the action potential from occurring. So in between those nerve fibers, you have inhibitory synapses. So there, the receptor binding causes the postsynaptic potential to be more negative or cause hyperpolarization, and that makes it more difficult to reach the threshold. And in fact, with both the excitatory and inhibitory nerve fibers, there's like different types of neurotransmitters that are being used. So an example of an excitatory neurotransmitter would be acetylcholine, whereas a type of inhibitory neurotransmitter, there's one called GABA. But those are just the extra details that you don't necessarily know. Now, another property of these action potentials, or the way these nerves are going to be firing, and that's going to be that blank right there, it's summation. So summation is when you have when you have two or more nerve fibers that are being transmitted at the same time. So two different excitatory nerves that are sub-threshold that are firing at the same time, they could add up. Those signals could add up to the point where it's high enough to pass that threshold and send an action potential. When you have a excitatory nerve that's firing an amount that reaches the threshold, but also an inhibitory nerve that's firing at the same time, the resulting signal may not reach that threshold and may not start the action potential. The next thing is called frequency. So frequency, it's intuitive what that word means. It's when you have firing and then quickly fire again, so a faster frequency. So if the first firing is sub-threshold and you fire again before the previous depolarization happens in that graph, then the new depolarization might be high enough. It'll be higher than the first time. It might be high enough to reach the threshold and then cause an action potential to descend. Okay, so now we'll talk about the way that the nervous system is actually structured. So that was what we just talked about. That was more on a microscopic level. It was more like, just the nerve cells and the action potentials. But now we'll talk about the actual nervous system as a whole. Obviously, the main functions of the nervous system, it's to exhibit the high-level control and integration of body systems, to respond to external influences, and you have sensory input and motor output. So sensory neurons, which I briefly mentioned earlier, they're also called afferent neurons with an A, and these are the ones that are you're getting signals from the body back to the central nervous system. Whereas the motor neurons, those are the efferent neurons. It's the other way around. You're going from the central nervous system to the effector organs. Hold on, it got really, really dark in here, so I'm going to turn on lights. Give me a second. Alright, so we got a little bit more light in here so it doesn't look this creepy. Okay, so now in this diagram you can see kind of how they're split up. So you have, the main distinction is between the central nervous system and the peripheral nervous system. So the central nervous system, it consists of the brain and the spinal cord, which are those blanks right there. And the peripheral nervous system, that's pretty much everything else. So in those blanks right below there, it's just what I had up at the top. Sensory, it equals the afferent part. Those are the nerves, like I said, carrying signals towards the CNS. Whereas the motor, those are the efferent. Those are being taken from the CNS to the rest of the body. And then within there, it's further split up within the motor neurons to somatic and autonomic nervous systems. The somatic nervous system, that's anything with voluntary control. So that's anything like with your skeletal muscles or anything that, like anything that you have control over is, I think, the best way to say it. Also, sorry, my room's a huge mess, but we'll just keep going and I'll clean up before the next class. The autonomic nervous system is the involuntary part of your nervous system. So that's going to be affecting all your visceral organs. Yeah, I know there's really nothing in my room except all of these clothes on the floor. So, yeah, it is pretty gross, isn't it? So, now we'll look at, within the autonomic nervous system, it pretty much breaks down again into two different parts of the autonomic nervous system. And that's going to be your sympathetic and your parasympathetic system. So sympathetic, that's going to be, your epinephrine. It's going to be your flight or fight response. And your parasympathetic, it's going to be rest and digest. So whenever you think of like let's say that you see a bear, the things that your sympathetic nervous system does, it makes your it dilates your eyes so you can see better. It increases your blood sugar so that you have more energy in your body. It sends more oxygen to your muscles. Whereas the parasympathetic, which is rest or digest, what happens there is it's going to be activating pretty much things like your stomach, or it constricts your eyes. Those are just some of the examples. Scroll down. Okay, so you have another distinction is your sensor and effector neurons. So the sensor, just like your sensory neurons that we talked about, those are carrying signals from your body to your CNS. So from wherever you get your senses, so like your eyes, your nose, it's sending smell signals. Your ears is going to be sending audio signals. Your sense of touch, it's going to be sending pain signals, for example, back to your CNS. Whereas effector, it's going to be the opposite. So this is going to be the motor signals going from the CNS to the rest of the body. Okay, so sympathetic and parasympathetic nervous system, so a little bit more detail here. The sympathetic nervous system, it's preparing your body for activity. So, I mentioned some examples earlier, but some more are: it increases your heart rate, it increases your blood pressure, causes more blood flow towards your muscles and less to the digestive system, causes your pupils to dilate, and it breaks down your glycogen to release glucose into your blood so you have more energy. Parasympathetic, it prepares your body for rest. So rest or digest. Decreases heart rate or blood pressure, less blood to the muscles, more to the digestive system. Pupils constrict, and it instead of releasing glucose into your blood, the glucose is actually stored as glycogen in your glycogen storage, which are both in your muscles and your liver. That's my roommate's alarm going off. I think he might be passed out. I'm going to go turn that off real quick. Sorry about that, but I think the thing is going to go off because I couldn't completely turn it off. I just snoozed it. Okay, where were we? Reflex loops. So when we talked about enzymes, we kind of talked about feedback loops in a pretty much more of in like a simple sense. So a feedback loop, that's when you have either positive feedback or negative feedback. Or you can also have a reflex arc. So positive feedback, that's when something causes something else to happen, and the result, it causes more of it to happen. So an example would be with uterine contraction. It leads to oxytocin release, which leads to more uterine contraction. Another example would be with blood clotting platelets. So the blood clotting platelets activate at a wound site, it attracts more platelet activation and more clumping. Negative feedback, it works exactly the opposite way. So you have an example is if you have blood pressure that causes ADH release, that is going to be increasing your blood pressure because I guess you'll learn about that hormone when Dr. Sutton talks about the endocrine system, but ADH is antidiuretic hormone. So what that does is it causes your body to retain water. So because you're retaining water, you're going to be increasing the volume of your blood. And when you increase the volume of your blood, you're going to have increased blood pressure. So the ADH, it increases blood pressure. So when it increases blood pressure, it causes a drop in ADH. So the increased blood pressure directly causes a drop in ADH. So that's negative feedback. Reflex arcs, those are like, whenever you're responding to a different a stimulus. So it's usually negative feedback. The example that we use is like a knee jerk. So when you tap the knee tendon, it causes a stretching of the muscle, and that leads to a contraction of the muscle. So it causes a knee jerk to happen, and that's negative feedback. And the way it works is your receptors, it's going to it's basically going to receive this sense from your sensory neurons. It's going to receive some sort of sense from your receptors. So like we'll just talk about like a knee jerk. It's gonna receive that signal from the receptors down in the neurons down there, and that's going to send a signal through your sensory neurons and eventually back to your integration integration center, and that would be your brain. And from there, your brain sends a signal back through the motor neuron to contract that muscle. The site of the stimulus, it's called the receptor. The thing that carries the impulse from receptor to integration center, it's right up in the little like pathway that's, I guess, just a couple lines above that. It's the sensory neuron. That's going to be carrying the impulse from the receptor to the integration center. And the next blank, it's integration center. So that's going to be connecting your sensory to your motor neurons. So that's a part of your CNS. You can have both monosynaptic or polysynaptic of those. In monosynaptic, there's no interneurons there. So the interneurons are the part of your integration center. So it goes directly from your sensory neuron to your motor neuron. Or you can have polysynaptic, where you do have interneurons there in the middle. Motor neurons, we've talked enough about that. So the effector, that's the site of the response to the stimulus. So I'm going to scroll down. Okay, role of the spinal cord and brain. The spinal cord, it's going to be providing the synapse for the reflex arc. And even though the reflex arc bypasses the brain, so I guess the integration center is not exactly the brain, but it bypasses the brain, but the brain is still aware of it happening, so it's still in control. Now, when you have efferent control, so that's when the brain it can actually override the spinal reflexes. So you can kind of control it a little bit. Wow, we're getting through this really quick. I don't think there's that much left. Yeah, there's only like a little bit over a page left. Okay, so now we'll talk about sensory reception and processing. Sorry guys, I'm still a little sick. So at your skin, you have proprioceptive and somatic sensors. This is where, here the first blank, it's going to be skin. So skin is where you can sense touch, heat, pain, and this is all going to be close to the surface, so right at that dermis-epidermis boundary. And there's actually pressure receptors that are deeper in the dermis as well. And the proprioceptor, what that does is it senses the position of a body part. So it can sense like, oh, my hand is here, or whatever. And those are specifically located in muscles and connective tissue. Somatic sensors, there's a number of different types. I'll type these out as I talk about them. So touch and pressure, it's sensed by mechanoreceptors. Temperature changes, those are sensed by thermoreceptors. Light is sensed by photoreceptors. Taste and smell are sensed by chemoreceptors.
Awesome.
And finally, pain, as in extreme heat or cold or pressure or different chemicals, that's going to be sensed by nociceptors. So those are the names of all the different receptors that you're going to know in terms of the nervous system. Now we'll talk a little bit about these different senses. This part is not like super, super important for the MCAT because I just, like in my experience, I haven't really seen it that much in passages and stuff, but you should still know it anyway because they say you're supposed to know it on there. At least the AMC says you're supposed to know it. The first sense that we're going to talk about is olfaction. So that's the sense of taste. So, oh, sorry, olfaction is actually the sense of smell, and then taste also works similarly. So we'll talk about them together a little bit. So with olfaction, you're going to have chemicals that enter your nose, but through your nostrils, get into the nasal cavity, and they get trapped in the mucus at the very top of the nasal cavity. And after that, they're picked up by the membrane receptors in the cilia and of the olfactory receptor cell, and that causes a cell depolarization and subsequent transduction of signals to the brain. So that's the pathway of how your sense of smell works. Your sense of taste also has to do with chemicals, so it's also involving the chemoreceptors. Chemicals get actually dissolved in your saliva, and they're carried to the taste buds. And at the taste buds, you have like hair-like microvilli, and those pick up the chemicals, and that causes the release of different neurotransmitters that send the signal to the brain. It's your sense of taste. Now, your sense of hearing, it's going to happen obviously at the ear. So a few things that you have to know about the ear. There's a couple of terms here that you have to know. So the ear canal, it's also called the auditory canal. And your eardrum, it's also called the tympanic membrane. And these are the ones that you kind of have to memorize. You have to memorize the different bones of the ear, because sometimes they bring them up. And there goes that alarm again. I'm going to go turn that off one more time. Let's get back to your workout. So, so the different ear bones. You have the malleus, the incus, and the stapes. So the common names for those are the hammer, the anvil, and the stirrup. But the ones that you need to memorize are the actual medical names: malleus, incus, and stapes. The vestibule, that contains the oval window where these ear bones, what they do is they pretty much vibrate, and that sends a signal to your brain, and that's how you hear. And the vestibule, this is where the stirrup, it vibrates, so the stapes. And it's continuous with the semicircular canals and cochlea. So all these different terms, I think the extent to which you have to know them is just know that they're a part of your hearing sense. They're in your ear. So like the cochlea, that's the little spiral thing that's in your ear, and that's where there's going to be hair cells there. And the semicircular canals, these are involved in your balance. So there's 3 of them that are all perpendicular to one another, and they pretty much sense the position and movement of your head, and they allow you to balance. And the actual mechanism of hearing, it goes like this. You have sound that enters the ear, then that hits the eardrum, which is the tympanic membrane, and then these different bones, the malleus, the incus, and the stapes, they pretty much vibrate, and the vibrates. That causes vibrations in the fluid in your cochlea, in your ear, and that transmits. That's a little bit unclear. It transmits to the fluid in the cochlea, and then the cochlear hair cells are excited by these vibrations, and they send a signal to the brain, and then you hear. Okay, so now we'll talk about vision. So this is through this is based on your photoreceptors that are located in the back of your retina. There's 2 different types of cells back there called rods and cones. So rods, what they do is they sense light and dark, so not color here. And these are a lot more sensitive. So if you look at the sun, you're overexciting your rods, and it's because they're very sensitive. The cones, those actually sense the different colors, and they're a little bit less sensitive. The chemical that's responsible for light reception, that's called rhodopsin. And this is basically retinol, which is a chemical, and opsin, which is a transmembrane protein. So together, it's called rhodopsin. And light is converted cisretinal to transretinal. So this is kind of like, I guess, organic chemistry will go over the difference between cis and trans, and it converts the chemical cisretinal, the chemical cisretinal to transretinal. The transretinal, it then causes the hyperpolarization of the photoreceptor cell. and that's going to cause a whole chain of events, a whole cascade that sends a signal to the brain. And that's going to happen through a bundle of nerves that's at the back of your retina, so at the back of your eye. And specifically, that's actually where the blind spot is. It's background music to the lecture. So the eye structure, I guess it's kind of, it says light first travels. Okay, so cornea is like the first thing in your eye, the thing at the very surface. That's where the light comes in through. And then it goes through your pupil, which is a hole in the iris muscle. The iris is the actual color part of your eye. Then it goes through the lens. So it goes cornea, pupil, lens, and then behind the lens you have all of the liquid part of your eye, which is called the vitreous humor. And then finally at the very back is your retina, and that's the screen that the image is kind of projected onto. And that's where the photoreceptors are. And that's where the rods and the cones are. Okay, visual image processing. So this relates to physics, and when we talk about different lenses and convex lenses and concave lenses and stuff, the lens of the eye is just like a convex lens in physics. So convex lenses, what they do is they form real images. So your lens, it forms a real image on your retina, which is the screen at the back of your eye. And real images are actually inverted. But even though the image on your retina is going to be inverted, the brain actually processes that image, and the way you perceive it is the fact that it's upright. Even though on your retina, I don't know if you can actually do it, but you might be able to. There might be things out there where you can actually see what is being viewed by the retina. It's going to seem inverted, but your brain makes you think that it's upright. And the brain, it actually combines two images from each eye in order to make it seem 3D. So it helps you judge the distance of that image. And one more reason why you have two eyes and you're processing two images is to get rid of that blind spot, because each eye in the very back over by the retina, there is a blind spot where all this stuff is actually processed. So that is all the material we covered in 45 minutes for the nervous system. Now we have the endocrine system, which you're going to go over with Dr. Sutton. But let's look through these notes, because I don't think he's going to use these notes, and we can fill in the blanks, and just so you have it for yourself to review later. So this first blank right here, let's see. That first blank right there is hypothalamus. The second blank, I don't know why it's not loading in my document right here. Give me a second. Let me open it up. Okay, so the second blank there is the pituitary. The third blank, or the one down here, when we're talking about the adrenal hormones, it's epinephrine and norepinephrine. Down here under pancreas, we're talking about glucagon. That's the one that increases blood sugar. The one that lowers the blood sugar is insulin. Okay, so hypothyroidism, that's when you have too little thyroid hormone or decreased thyroid hormone. And hyperthyroidism is when you have increased thyroid hormone. Okay, so see. The glands that are directly respond to chemical levels in the blood, those are called humoral. Humoral. The next one is neural, Neural. And that final one over there is hormonal. And that's the whole document. Alright guys, so do you guys have any questions over nervous system? Because we still have like about 10 minutes before Dr. Sutton will take over for, yeah, what's up? Yeah, sure. So the first one is humoral. Yeah, I can actually, I'll just type them out for you. Okay, so those are the last ones. Let me see if there's anything that I could talk about in a little bit more detail. Nope, that's pretty much it, guys. I guess we have 10 minutes. Go ahead and take a break, and at 7 o'clock, Dr. Sutton will be back on here, and he's going to talk about the endocrine system. And I'll probably peace out. Ten minutes just to make sure that he gets the whole thing started, and then I'll go. So I'll meet you. Yeah, so y'all just come back to your computers at 7 o'clock. So right now, in terms of set status in that top box, change it to away or step away. So I know you guys aren't by your computer or whatever, and then when you come back, you can change it to I guess get rid of the step away part. The opposite of insulin is glucagon.